today I would like to finish the block of the neurohistology lectures, and this talk is about the glial cells. In the upcoming 45 minutes, I try to summarize the most important points to figure out what the main functions of the glial cells are, and I have to put some emphasis on their function and structure as well. So in the beginning, I just would like to remind you that the nervous tissue is made up by nerve cells and glial cells, and I will classify the glial cells. Then we will go step by step and discuss briefly the peripheral glial cells. I will focus more today on the centroglial cells, where we will define two main groups. One that contains this GFAP protein, another group that does not have this protein, and within each main group we have several members, astrocytes, including protoplasmic and fibrous ones, Ependymal cells, Bermudge cells, Pitohicites, and Müller glia. And to the GFAP negative group, we include the oligodendroglial cells and the microglia. This is what I would like to talk about today. And I would immediately start with the cells of the central nervous system. This is not a new slide. You must know this already, that the nervous tissue is made up by two main families of cells. We have the nerve cells or neurons. This is what we did in the last lectures. I told you that their main function is to control the widest biological variety of biological functions in the human body, because they are specialized for the transmission and the processing and relaying of the electric signals. And this they do through synapses. This is what we did in the last lectures, and we are done with neurons, but we didn't discuss yet what are those cells which will provide, which will help, which will provide aid for the nerve cells. And this is the second family of the glial cells, and this is the second family of the cells in the nervous tissue, and this is the glial cells. And this is what I will talk about today. The glial cells receive their name from the original idea that they would just fill the space between the neurons. People knew already in the early era of neuroscience that these cells the neurons should have the main function. But now we know that without glial cells, the neurons couldn't perform their action. So we know now that they are much more important than just filling the space, like the glue would fill the space between two objects if you want to fix them together. So we can find them in the central and in the peripheral nervous system, and besides their mechanical support, they also have several functions, as you will see. We have central and peripheral groups, and the peripheral ones you already know more or less, but I will come back to these very briefly. So don't forget that the nervous tissue is made up by nerve cells. They carry the most important part of the function, but without glial cells, they don't work. They just don't survive. Therefore, glial cells are also essential. If we want to classify the glial cells, one can say we have the peripheral ones, and this is the Schwann cells and the satellite cells. Already I mentioned that, and when I discussed the synapses and the effectors with you, I mentioned that in the receptors and in the effectors we have some specialized types of glial cells. I mentioned the teloglia, for instance, in the neuromuscular junction, if you can recall that, but other nerve endings like the receptors also have specialized glial cells. I don't want to discuss those today in detail, but what I have to talk about is the central glial cells. If I want to discuss the centroglial cells, nowadays people follow the classification according to the fact if these cells contain a specific protein called GFAP. It comes from the short abbreviation of the glial fibrillary acidic protein, which is a type 3 intermediate filament that is involved in the cytoskeleton of the glial cells. Not all the glial cells have it, but we have a certain group of glial cells which is GFAP positive. And if you perform an immunolabeling for GFAP, you can identify these cells, so one can use them for distinguishing these cells. It's a histological marker at the same time. So there is a certain group of centroglial cells which are therefore negative with this marker, so they do not contain GFAP. So these two main groups one can define within the central glial cell family. And if I start with the GFAP positive ones, we have the astrocytes. Two subgroups have to be defined, fibrillary and protoplasmic astrocytes. They are found in the white and in the gray matter respectively. I will come back to these. I will have multiple slides about these cells. Ependymal cells I will also highlight. Bermudge cells belong to this group. Pitohicites belong to this group. They are found in the pituitary gland, in the neurohypophysis, as you will see. And finally, we have the Müller glial cells. They are in the retina. GFAP negative glial cells are the oligodendrocytes. Also, the microglial cells belong to this group. Please note the main function I also indicated in the classification, just always one point that you can link it to something. Oligodendrocytes are responsible for the myelin sheath formation in the central nervous system. Microglial cells are the macrophages. Here the terminology is a little bit fuzzy. People use sometimes the term mesoglial cells. That's because of their developmental origin. I will come back to this. Or the eponym for that, Ortega's glial cell. This was the scientist who first time published these cells. So this is the classification of glial cells, and if you open different books, you will find different classifications. In order to avoid misunderstandings in the exam, I highly recommend to use this classification, because this is what our department wants to hear back in the exam, in contrast to older classifications which are outdated already. So let's go on and let's talk a bit about the peripheral glial cells, just very briefly to review those key points what you already know about that. So some hints on the peripheral glial cells. Schwann cell, you know this already. I discussed with you that they form the myelin sheath or they form the Schwann sheath. You can recall this most likely that the axons will be invaginated into the cell membrane double layer that is wrapping around the axon, and this way you get this myelin sheath, or they are just covered by the cell processes. They hug around the axon, and this is how the unmyelinated fibers are formed. This is the Schwann sheathed structure. Some more details to add. These cells are also positive GFAP. So, however, they are in the periphery. One can identify them by GFAP AP immunolabeling, and with the electron microscope, one could also see that there is a basal lamina outside the Schwann cells, lamina basalis, if you wish. You can use the Latin name as well. Ion channels are found in the cell membrane, which are responsible for the ion transport processes, and during the embryonic development and also later, the axons functions are controlled by the Schwann cells, and the other way around, the axons will also influence the functions of the Schwann cells. So Schwann cell axon interaction exists, and the neuroregulins and the cytokines are molecules which are produced in part by the Schwann cells, and they are therefore involved in the control of the axon diameter and also growth and axon transport. I mentioned when I discussed with you the regeneration of the axon in the periphery that growth factors are also produced by these cells, which will then control the regeneration as well. And also during the development, if no injury is there, but the axons just are newly developing, the proliferation and the apoptosis of the Schwann cell is depending on that if the axons are there or not. So the axons and the Schwann cells will interact with each other, and they control each other's development and function also. Embryologically, you will see that the peripheral nervous system is derived almost completely from the neural crest. This was also true for the Schwann cells. Satellite cell is the other type of the peripheral glial cells. You remember this is something that we met in the ganglia. This week you will review the histology of the peripheral nervous system, and the ganglion slides will come up, and there you will see the satellite cells around the big neurons. They are responsible for the nutrition and the support of the neurons, and they also contain glial fibrillary acidic protein, which helps to identify them. And the basal lamina is also there, so you can find many similarities with the Schwann cells, which is true for the ion channels and for their developmental origin also. So these cells are not only similar, but also are derived from the same primordium, from the neural crest. So there are several similarities between satellite cells in the ganglion and the Schwann cells, which forms the myelin sheath. So please, if you have to talk about the classification of the glial cells, at least mention the peripheral glial cells. And when you review these slides, take one moment to collect the main important points. And from now on, we would like to go on with the centroglial cells. And once again, that you remember well, so we are here now. The peripheral is done. And we turn now to the centroglial cells, and I will go on with the GFAP positive population with astrocytes. And in the next slide, I compare the protoplasmic astrocyte with the fibrillary astrocyte. You will have a preparation stained with gold sublimate in the classes in 2 weeks. You will study this slide, and there you can compare the protoplasmic and the fibrous astrocyte in the same preparation. Also, you can see the same cells with Golgi staining in other slides, in the cerebellum, for instance, or in the cortex slides from the cerebrum soon. The protoplasmic astrocyte is found in the gray matter. In contrast, the fibrillary astrocyte is found in the white matter. So if you look into the microscope and you can distinguish the white matter from the gray matter, and you see astrocytes, based on the localization, you will know already which one is the protoplasmic, which one is the fibrillary. But there are also some other differences which, however, require some practicing in order to be able to distinguish. So fibrillary astrocytes have more tiny, longer processes, and they will then, compared to the protoplasmic ones, are quite different, because they are, in the case of the protoplasmic astrocytes, fewer, but they shorter, and they are much more strongly ramified. So you see it's like a bush or something. It's a really typical round, very tiny, several times ramified processes. Also here you can compare a protoplasmic with a fibrous or fibrillary astrocyte. The difference is, I think, obvious. Because the cell body part is larger in the case of the protoplasmic astrocyte, it has much more stronger developed intracellular organelles than you can find in the fibrillary astrocytes. The astrocytes produce or contribute at least to the limiting membranes. In the upcoming slides, I will come back to this, but I will talk briefly about the blood-brain barrier, about the brain surface and the subependymal layer. And the protoplasmic and the fibrous astrocytes both contribute to these surfaces. In the white matter, the fibrillary astrocytes form the blood-brain barrier. In the gray matter, we have also these surfaces, like on the surface of the brain, on the top of the cortex, on the gray matter, we have a border, a limiting membrane, and also around the vessels, we have the blood-brain barrier. But I will come back to this point. And finally, this is not a difference. This is their common feature that intermediate filaments made up by GFAP is found. So you can use this molecule as a marker for the astrocytes. So if you want to compare the astrocyte subtypes, protoplasmic and fibrous astrocytes, fibrillary astrocyte, you can use this slide. Then the astrocyte functions I would like to discuss in the upcoming slide. And this slide is about the limiting membranes, at least in part what I was mentioning in the last slide. So you have the surface of the brain here. And on the surface of the brain, you can see that the astrocytes will approach the surface with their processes, and they have a flattened end part, which is called end foot or end feet, and they will then cover the surface of the brain. Then a basal lamina is coming, and the pia mater is coming, which is blue here, and then these layers will together form the surface, the external limiting membrane, and this the main layer of that is the as astrocyte and feet layer. Now, please note that the pia mater also goes on, and sometimes where the blood vessels enter the brain, they like a funnel go into the deep, and they sometimes leave a space open, which is the so-called Virchow-Robin space. This will have later significance in the imaging technique. Sometimes you can see these as little holes in the brain if you cut a horizontal section of the brain. This is related to the meninges also in the corresponding lecture. It will also come up later. Then you also have another limiting membrane, which is again, at least in part, formed by the glial cells. So if you follow the blood vessels, they of course split, and finally you get the capillaries. And the capillaries have, of course, a wall that is made up by the endothelial cells. Then you have a basal lamina with pericytes eventually, but they do not form a continuous layer. And then we have a continuous layer of astrocyte and feet again. And these layers together will form the deep or internal perivascular glial limiting membrane, also called blood-brain barrier. We nowadays think that the endothelial layer has the greatest significance in the blood-brain barrier, but also the astrocyte and feet will provide a layer for this blood-brain barrier, and ion channels and transport processes and complex processes will take place here, which has a great clinical significance, and this is what I'm mentioning here. So if you want to influence the brain's function with medicines, for example, you have to create a molecule that can pass through through this barrier in order to enter the brain. But if you would like to give a medication and your goal is that that does not enter the brain because it would cause side effects, for instance, you have to create a molecule that does not pass through the blood-brain barrier. So in pharmacology, you will talk a lot about the function of the blood-brain barrier and how medicines are distributed within the central nervous system. In this figure already it's mentioned that the astrocytes can also pick up neurotransmitters from the synaptic clefts, as I mentioned earlier when we were talking about the synapses, that glial cells will remove the neurotransmitters from the synapse. Another function of the astrocytes, what I have to define, is the nutritive function, and that's also a vital thing. So blood vessels we have, they are surrounded by the end feet of the glial cells. So the nutrition that comes from the bloodstream, the glucose practically, has to pass somehow, and this is something that is helped by the astrocytes. So there is a quite complex biochemical breakdown here, And I don't want to go into those details, but you know about the biochemistry a lot. So these glucose molecules are picked up from the blood and in a form of lactate and other molecules, it will be then transported from glial cell to glial cell. And they may be, the glucose may be stored as glycogen for a shorter period of time. The lactate is transported to the neurons, and this is how the neurons are fed. And importantly, the lactate can also be transported into the oligodendrocytes, which then finally will feed the axons. So in different aspects, the astrocytes have a great significance in the nutrition of neurons, and without this action, the neurons cannot perform their function. So this is what I started with today, that we have the glial cells, we have the neurons, and indeed the neurons have the more important functions, but they can't do anything without the support of astrocytes and other glial cells. In this slide, I made a list about the functions. The limiting membranes I mentioned already, the nutrition I did also. What I didn't mention yet was the establishment of the extracellular matrix. This is something that I didn't mention yet, that within the central nervous system we have, however, a very narrow, but we do have a narrow extracellular space that is somewhat similar to the extracellular matrix of the connective tissues, but we do not have really strong fibers here, but we have instead these extracellular matrix molecules like heparin, sulfate, proteoglycans, laminin, fibronectin. They are also found in the central nervous system, and this is also a product of the astrocytes. What people already identified in the early era of neuroscience, and what the initial idea was that the glial cells have a supporting function, is still true, but this is not only their only function, but they still provide the support, and they separate the processes of the cells from each other, which is also important in the communication. I mentioned the breakdown of neurotransmitters, as I showed you in an earlier slide of mine, that the synapse might be considered as a synapse that has three parts: presynaptic, postsynaptic part, and the glial cell would be the third party. So that's why tripartite synapse can be also used as a term for that, that the glial cells will clean up the neurotransmitters from the synaptic cleft. I also mentioned that the electrolytes, potassium and calcium ions, may be stored in the glial cells, and during the embryonic development and during nerve injury also glial cells may influence the development, the growth, the regeneration by producing growth factors. They also produce cytokines, influencing the inflammatory processes, and in many biochemical processes, redox processes are involved. Hydrogen sulfide is produced also, which is a gas neurotransmitter. Lactate I mentioned, related to the nutrition. So there are several biochemical issues which come up if it's about astrocytes. This is also characteristic for the astrocytes, that they can divide. If there is a stronger tissue damage, they will start to grow, proliferate, and they even may get activated that far that they start phagocytosis. This is not their main function. Phagocytes are the microglial cells. This is what I will finish with today. But they produce also some scar tissue. After a tissue damage, they clean up the dead neurons, and after that they stay there and they produce a glial scar tissue, which is called also gliosis in the histological terminology. And you most probably heard about a specific kind of cancer that is coming from the astrocytes, the gliomas, or in the worst case, the glioblastoma, which is a very fast-growing, very bad malignant disease, which will grow in the brain, and this is nowadays a cancer that we cannot really cure. We treat it, we can win maybe some time, but this is a very bad diagnosis because there is practically no cure for that. We just can win some time. All right, after the astrocytes, I would turn to the ependymal cells. Just one slide about that. Ependymal cells are found in the brain and in the spinal cord. They line the cavities which are filled by cerebrospinal fluid. This is the brain ventricles and the central canal of the spinal cord. They are sometimes cuboidal, sometimes columnar cells carrying cilia on their surfaces. They might be in part kinocilia, in part microvilli. And they also have connections between each other, electric synapses, gap junctions, and mechanically they are connected by desmosomes, belt desmosomes around the disc plane. And they may have basal processes which grow into the nervous tissue, sometimes even far away. If these basal processes are really long, one can call them tonycytes. And these tonycytes are found in the hypothalamus, for example, in humans also. This is a rat hypothalamus, but it could be a human also. We have also those cells there. And it has been shown that these cells have regulatory function because they are connected to the cerebrospinal fluid, to the nerve cells, and to the blood vessels, and they can monitor the blood composition. They can also measure what happens in the cerebrospinal fluid, and they have therefore a regulatory function. I indicated here the GnRH. This is a hormone, you might be familiar with this, that controls the gonadal functions. And tonycytes have an impact on the GnRH release from the hypothalamus, but one could give several other examples about this GnRH. You will hear a lot later when we talk about the endocrine systems. Also, the ventricles may contain a structure that is responsible for the production of the cerebrospinal fluid. That's the choroid plexus. And the surface of the choroid plexus is covered by a similar epithelium, which is very similar to the ependymal lining of the ventricles. Okay, the next cell, what I have to mention is the Bermudge cell. This you will meet when you talk about the cerebellum. And the cell is a type of protoplasmic astrocytes that is GFAP positive, and they have a little bit weird function, so they are not round and they don't have in all directions ramified processes, but their process goes perpendicular to the surface of the cerebellum. That's why some authors call them radial glial cells also, because of this radial pattern of their processes. And besides the typical astrocyte functions, we know about the Bermudge cells that during the embryonic development, the granular cells of the cerebellum, which are located in this deeper layer, will migrate along these cells. They just go down along these processes because these cells develop on the surface almost, and they migrate downwards. And this migration takes place along the processes of the Bermudge cells. They have therefore an embryological significance also. Then we turn to the Pitohicites now. The Pitohicites are again modified cells, modified astrocytes. One can find them in the pituitary gland. More specifically, we have in the pituitary gland this posterior lobe, which we also may call neurohypophysis. And in this neurohypophysis, if we perform a higher magnification imaging, one can find these kind of nerve fiber enlargements called herring bodies. And around them, one can find these larger cells, and they are the pitohicites. And the herring bodies are responsible for the storage of those hormones which are released from the neurohypophysis. You will get lectures about that. I don't want to explain you everything. I just have to talk about the glial cells today. But the hormone release from these herring bodies goes into the blood vessels, which are found here. These are specific sinusoid capillaries. And the substances, the hormones which are released from the hearing bodies will go into the blood vessels if the pitohicites pull back their processes. If we don't need that much hormones at a certain moment, then the pitohicite processes will envelop, will surround the hearing bodies and kind of seal them. And then if we need the hormone, they pull back their processes, and then the way is open towards the sinusoid capillaries, which can then pick up the hormones. This is how they work. And also they have a mechanical supporting function on unmyelinated fibers, and they are involved in neurotransmitter in glutamate uptake, and they also pick up from the extracellular space the potassium ions. So they have typical astrocyte functions, plus their specific feature is that they are involved in the control of the hormonal release that is specific for the neurohypophysis. Astrocytes called pitohicite. And the final centroglial cell that is GFAP positive is the Müller glial cell. And you have studied the retina already, so this is just a recap now. You know the 10 layers of the retina, and you also know that there are limiting membranes. There is an internal and an external limiting membrane formed by the Müller glial cells. Their end feet will form these limiting membranes, and the cell body of the retina with the nucleus is located in the internal granular layer of the retina. You remember there were neurons like amacrine cells, horizontal cells, and bipolar cells, and also a glial cell, the Müller glia was there. A few years ago, it has been invented that the Müller glial cells, besides the typical astrocyte functions, have also very funny and very tricky significance, namely that they will help to conduct the light towards the cones, because they work as fiber optics. Like the light beam comes into the retina, and you know that the human eye has an inverse retina, which means that the light beam has to cross many layers before it arrives to the actual photoreceptors. Therefore, something has to aid the light conductance, and this is done by the Müller glial cells, because they will carry the light towards the cones, and this way they can be activated. They are also GFAP positive cells, but the retina is not new for you anymore. So when you review the retina, please make sure that yes, there was also a specific kind of glial cell which has to be inserted into the classification of the glial cells. All right, we are done with the glial cells of GFAP positive type, and we turn now to the GFAP negative glial cells, and these are also quite important. There are some books which call the GFAP negative glial cells as an upper term microglia because they are small. But be careful with this terminology, because the oligodendroglial cells are a specific entity, and we also have microglial cell proper. So this kind of classification is somewhat misleading. Therefore, I would suggest to skip it and say instead we have the GFAP negative glial cells with two groups, oligodendroglial cells and microglial cells. In some books you will find this classification. It is already outdated, and we don't suggest to use it anymore. So what to know about the oligodendrocytes first? Oligodendrocytes, as I mentioned, are smaller cells than the others I discussed until now. They have less processes than those. Importantly, if you stain them with hematoxylin eosin, they have a very basophilic, heterochromatic, almost black, very dark nucleus. Now, the processes will be the most important ones, because they will approach and they will surround the nerve fibers, the axons running in the neighborhood, and they have a very similar function to the to the cells in the periphery, which form the myelin sheath. So they have, in function, similarities with the Schwann cells, but they are now in the central nervous system, and therefore they are the myelinating cells inside the brain and inside the spinal cord. The most important difference, if you compare it with the Schwann cell, is that they have a process that approaches a certain axon. It wraps around it, but at the same time, the same cell may send multiple processes to other axons. So one cell may have many processes, each may contribute to the myelination of multiple axons. So up to 50 axons may be myelinated, at least in part, of course, by one single oligodendrocyte. And this is an important difference in comparison to the periphery, and where one Schwann cell forms in the myelin sheath only for one single axon. Now, you might come up with the question, why is this? The question, or the answer to this question is quite hard, but people think that if we would have individual glial cells for all our axons in our head, the skull or the human brain would be extremely huge. I don't know how people could figure this out, or how this calculation was done, but I read somewhere that the diameter of the human head would be around 1.5 to 2 meters if we would have Schwann cells inside the brain instead of oligodendrocytes, so they spare space. But of course, if we spare, we always sacrifice something, because we spare space. These cells also have disadvantages. Missing basal lamina is maybe not a disadvantage yet, but if the process or if the axon is damaged, and for a while the axon disappears because it is cut, then the oligodendrocyte will pull back its process, it retracts the process, and the axon will not find a way to regenerate, because the demyelination, the damage of the myelin, will produce a tissue debris which induces a gliosis, astrocyte proliferation, and a scar is formed, and therefore the axon cannot grow back into the original myelin sheath, in contrast to the periphery, where the regeneration of the axon is possible. So therefore, if the oligodendrocytes are damaged, or if the brain tissue is damaged, the axon cannot grow back to the original destination, and therefore the function is lost. Also, this myelin sheath somehow wakes up the interest of the immune system, and we don't know why, but in many cases, unfortunately, an autoimmune disease is initiated in which the myelin sheath is damaged, and this is what we call multiple sclerosis, which is a demyelination disease. Depending on the connection, depending on the anatomical localization, what connection of the brain is interrupted by the damage of the myelin sheath, quite variable neurological symptoms will occur. Unfortunately, this is a quite common disease which starts in young ladies most of the times, and there is no cure for that. We can treat it, we can give a quite acceptable quality of life for these patients for a longer period of time than earlier, but unfortunately we cannot cure the disease at the moment yet. And then finally, I would like to talk about the microglial cells. Microglia is a topic that that could be an independent lecture. So the microglial cells look very different from the earlier discussed glial cells. If you stain them, you see a very, very small cell body with extremely long, very tiny and multiple times ramified processes, which will form a certain framework or a certain network inside the volume unit of the brain. And this cell is inactive. This is how they look like if they are just resting. And If, however, something happens, for example, an inflammation occurs or a head injury occurs or something comes up that would damage the brain tissue, or just some inflammation is initiated, or a very strong activation of a particular brain area is already strong enough to induce that, the cells get activated and their morphology changes then. What you will see that they retract their processes, which become thicker. They will their surface will be a little bit uneven. It's a bit fuzzy, and you will see that the cell body will be also enlarged. And what you don't see, but you must know, that they start to produce inflammatory cytokines, which then will control the inflammatory response in a tissue damage. And if there is a very strong tissue damage that causes even neurodegeneration, so it's not only some demyelination or some tissue debris produced, but neurons will really die, and a higher amount of tissue debris is produced, then they get reactive, which means that they get even shorter processes. They get kind of spindle shaped with short processes, and they will start to perform phagocytosis. This is their main function. They are the phagocytes of the brain. And in this stage, they can already migrate, so they can move inside the brain. They can divide, and they will clean up, and they will remove through their phagocytotic activity the damaged brain tissue, and they will clean up. So these are the macrophages of the central nervous system. And you most likely heard about that macrophages are coming from the monocytes, and you have studied in the earlier semesters already the mononuclear phagocyte system. You have studied the Kupffer cells in the liver. You heard about the alveolar macrophages in the lung. You most probably heard about the osteoclast cells in the bone. So these cells, which are responsible for cleaning up, for breaking down something, are most of the times from the mononuclear phagocyte system, from monocytes. And this is also true for the microglial cells. So they can enter the brain. And this happens already in the embryonic life. So the microglial cells are there. They are resting cells. But if they get activated, then they start their phagocytotic activity. You will see slides about this in the class. One will be an immunocytochemistry, where we perform the staining on inactive and on active cells by marking the complement 3 receptor on their surface. This is the surface marker CD11b or beta, which is a surface marker, and one can produce antibodies against that, and you can show the microglial cells specifically with this marker. But if you are in the PH University and you study with us, I have to mention this silver impregnation technique, which is invented by Professor Gajas, who is Professor Gajas the senior. He's the father of the head of the biochemistry department. Professor Gajas was a very famous neuroscientist, and he is worldwide known about his silver impregnation techniques. And one of them was directed to identify the microglial cells. And we were very proud that we were working together with him, and he passed away last year. He was really famous on his technique, and if you go anywhere in the world and you speak to people who know something about neuroscience, they will know the Gajas technique. Maybe they cannot pronounce his name, but they know the name, and this is something that was invented in PH, and we are very proud of that. So the microglial cells can be shown with his silver impregnation technique, and this is also still used in neuroscience to show active phagocytotic activity in the cells. His technique shows the iron content of these cells, because they perform phagocytosis and because they remove the erythrocytes containing hemoglobin, which contains iron. One can show the iron content, and silver intensify the iron content of the cells, and this is how he managed to show these cells. All right, the time is almost over, so anyways, I would like to show you something that we did in the lab, and this is what I would like to finish with. This is a staining method that we performed in our lab on neurons, and we are busy now with Parkinson syndrome research, and we are interested in what cells do degenerate in the brain, and these neurons have to do with some mood control. And what we did, that we stained the microglial cells in the red brain after we made them Parkinsonian. And it turned out that some of our green neurons get also red, which means that they are attacked by the microglial cells. And if we zoom in a bit with a confocal microscope, one can see that this cell is falling apart, so we think it is degenerated. And you can see the red microglial processes as the dying cell is cut into parts, and it's removed by the phagocytotic reactive microglial cells. And I would like to thank for your attention, and because it's International Women's Day, this is for the ladies for their attention also, and thank you very much. If you have any kind of questions, just let me know, and I try to answer them.
I had a question on, you said multiple sclerosis has no cure, and you said, but you could treat it. How do you treat it?
Yes, well, you have several techniques to treat it. One option is that you do a so-called immunomodulation, which means that you perform you give a certain therapy using corticosteroids to push down the immune system a bit. This is in the acute case, and if you manage to stop the acceleration of the immune reaction, you can use immunomodulatory medications. These are cytokines, for instance, interferons, which then will influence the activity of the immune system, and you can slow down the reaction. But the disease comes and goes, so you will see that patients have a so-called attack or shub, which then causes new symptoms. You give a treatment, it gets better. And a few months, a couple of years, he is okay again, and then some new symptoms come again. Then you give a treatment, they almost completely disappear, and then it comes again. So the symptoms come and go. But after a treatment, usually you cannot restore completely the original function. You can get back, because some remyelination may occur if the axon is not damaged, but complete full remission is rarely achieved. If you manage it, you can be very happy about that, but usually step by step, the general condition of the patient gets, after each worsening, always a little bit weaker. And after 15, 20, 25, 30 years, for sure, more and more symptoms will stay. But this is what you can do, that you slow down each attack of the immune system by corticosteroids and by immunomodulatory treatment. You can nowadays use antibodies also, which are directed against T cells. You have many new therapeutic achievements, but we cannot really treat the disease in a way that we kind of reverse the story, because we don't know why does that happen. There is no point. It's not known yet why the disease occurs. We know some factors, but we don't know yet.
Okay, thank you so much.
You're welcome. Other questions, maybe? Okay, for those who are still here, because I ran out of time, I didn't want to show that, but maybe if some of you is interested, I have a video on this cell, what I showed you in my last slide. It was the degenerating neuron. This is what we did also in our lab. you can see the same neuron falling apart, and you see how the processes of the microglial cells approach that. And in 3D, you can see that from behind, above, the neighboring microglial cells will remove the dying green neuron. So I show you this because if any of you would have some interest on neuroscience, neuromorphology, those people who would be interested, we could talk about it. And some people may be interested in that and could join also the lab for some research, just in case. Okay, people, thanks for your attention. If no more questions, then I would say goodbye. Any remarks, questions?
Sir, I have one question.
Yes.
Can the sclerosis and the multiple sclerosis can lead in the future to amyotrophic lateral sclerosis or no?
The amyotrophic lateral sclerosis is also an autoimmune disease which attacks the motor neurons, and that has a different background, and it's a different disease. It's more rare. I cannot say at the moment if somebody who has multiple sclerosis may also develop amyotrophic lateral sclerosis or not, but it's maybe likely, but there is no, as I know, there is no real link between the two diseases. So somebody may be very unlucky and may probably develop both, but this is not very likely.
Okay, thank you, sir.
You're welcome.